Imagine an AI that doesn't just simulate human thought, but lives, built from actual human neurons, learning, adapting, and evolving like a biological brain. This isn't a scene from a sci-fi blockbuster, it's real. Meet the CL1, the world's first organic computer, a groundbreaking fusion of living brain cells and silicon, pioneered by Australian startup Cortical Labs. Unveiled on March 2nd, 2025 in Barcelona. The CL1 isn't just another step in AI evolution. It's a quantum leap, promising unmatched adaptability, energy efficiency, and learning potential beyond anything we've seen before. But what does this mean for the future of technology, and for us? Let's dive into the mind-blowing reality of synthetic biological intelligence, SBI, and its implications for the world. The CL1 biocomputer is not your average supercomputer. CL1 makes use of lab-grown human neurons, which are cells that can learn and adapt in real time, as opposed to conventional silicon-based AI processors that need large datasets and processing capacity that consumes a lot of energy. Its versatility and efficiency are just unmatched by conventional AI because of this. A network of living neurons contained in a life support unit that maintains them alive and working on a silicon chip is the basis of this technology. Similar to a human brain, these neurons interact, adjust to inputs, and become intelligent. The ability of human and mouse neurons to comprehend and react dynamically to information was first shown in 2022 when Cortical Labs' dish brain technology enabled them to learn how to play Pong. This idea is now transformed from an experiment into a fully working commercial product by the CL1. CL1 is revolutionary for the following reasons. Wetware as a Service, WAAS. The CL1 biocomputer is accessible for purchase or remote cloud access, enabling widespread use of biological computing. Self-learning and evolutionary AI. SBI learns in real time and continuously boosts its own efficiency, in contrast to standard AI models that need large datasets. Unmatched energy efficiency. Compared to the supercomputers that operate today's AI models, a single CL1 uses a lot less electricity. Next-gen robotics and drug discovery. SBI's flexibility has the potential to transform domains such as neurological research, customized medicine, and robotic intelligence. The CL1 is expected to democratize synthetic biological intelligence and pave the way for previously unheard-of breakthroughs, with a $35,000 price tag that is far less expensive than existing sophisticated computer systems. A network of lab-grown neurons derived from induced pluripotent stem cells, iPSCs, which are basically blank slate human cells with the ability to differentiate into any kind of cell, form the core of the CL1 system. These neurons may establish connections and process information similarly to a human brain since they are positioned on an array of electrodes that stimulate and monitor activity. The CL1's neurons self-organize, forming novel routes to solve issues in ways that even its creators cannot completely foresee, in contrast to conventional AI that functions with strict programming. One of the most significant developments in AI history is this organic intelligence. If you enjoyed this video, don't forget to like, share, and subscribe for more updates on the future of AI. Let's explore the future together. Moving on. In 2022, the group showed how human and rodent-induced pluripotent stem cells, iPSCs, integrated into complementary metal oxide semiconductor, CMOS-based high-density multi-electrode arrays, HDMEAs, could be electrophysiologically stimulated to create highly effective, autonomous information exchange pathways. To do this, scientists required a mechanism to penalize the brain cells for failing a task and to reward them when they displayed desirable behaviors. Predictability was shown to be crucial in the dish brain studies. Neurons would adjust their networks to find connections that provide predictable, energy-efficient results, while avoiding actions that result in a chaotic, unpredictable electrical output. However, Kagan said, the current version is a totally different technology. The prior one made use of a CMOS chip, which essentially provided a very high-density reading, but it was opaque, making the cells invisible. There were additional problems, such as the inability to draw out or balance the charge when stimulated with a CMOS chip. When you stimulate for extended periods, you end up with a buildup of charge, which is not good for the cells. These versions have a much simpler technology, but that means they're much more stable, and you can balance that charge much more actively, Keegan said. You can draw out 2 microamps of current when you put in 2 microamps. Additionally, you can prolong its stability. The Minimal Viable Brain, MVB, a synthetic neural system that mimics the functioning of the real brain but is tailored for computers, is the next project Cortical Labs hopes to construct. To find the smallest, most effective biological brain that can process complicated information, scientists are experimenting with various neuron kinds and configurations. This study has significant ramifications for neuroscience, advancing our knowledge of consciousness, brain function, and even illnesses like depression, epilepsy, and Alzheimer's. Researchers may simulate human brain activity with CL1 without involving humans or animals in intrusive or immoral experimentation. Chief Scientific Officer Brett Kagan examines a few lab-grown stem cells. Atlas new lab-grown neurons are positioned on a planar electrode array, or basically just metal and glass, as Kagan put it, within the CL1 system. Here, 59 electrodes serve as the foundation for a more reliable network, giving the user extensive discretion over when to activate the neural network. After that, this SBI brain is installed in a rectangular life support unit and linked to a software-based system for real-time operation. The perfusion circuit component serves as the cell's life support system. It has pumps to keep everything moving, temperature control, gas mixing, and waste product filtration. According to the team's press statement, a complete rack of CL1 units delivers bidirectional stimulation and read interface tailored to enable neural communication and network learning, is fully programmable, and consumes only around 851,000 W of energy. Surprisingly, the CL1 device can also function without an external computer. The CL1 units, which are designed to preserve the health of the cells residing on the silicon hardware Atlas, are being tested by Kagan and his colleagues. First and foremost, the intricate dynamic SBI neural networks, which, when seen under a microscope, seem to develop branches from electrode to electrode, have the potential to completely transform the fields of drug discovery and disease modeling. Although the second technique, known as the small molecule approach, yields a variety of cell populations, it's often unclear precisely what they're dealing with. For Cortical to continue its ambitious goal of creating the minimal viable brain, this must be understood. The team is working diligently on the next phase of SBI, even if the CL1 launch is the first step. How to bioengineer a human-like brain with the least amount of unnecessary cell differentiation, while yet achieving the complexity that developing a neural network composed of homogeneous cell types lacks, is an interesting idea known as the MVB. This kind of tool would be a potent model that would enable much more control and sophisticated analysis than are now achievable in actual brain research. According to Fundamental Ideas, it would basically be the key biological components that allow something to process information dynamically and responsively, Kagan said. A single neuron is capable of many things, but it is not able to traverse an environment or react to any degree of dynamic activity. The CL1 elegans has 301 or 302 neurons, depending on who you ask, making it the smallest functioning brain we are aware of. However, those neurons are exceedingly specific. Since AI has advanced so drastically, ethical issues will inevitably arise. Does a human brain cell-based AI have consciousness? Could it learn to think for itself? Furthermore, how can we distinguish between a machine and a live thing? According to Cortical Labs, CL1 is only a biological processing unit that has been educated to solve problems. It is not sentient. However, ethical and legal frameworks will need to adapt as this technology develops. To guarantee appropriate use of SBI, the business is actively collaborating with government organizations and bioethics committees. The CL1's unveiling marked the beginning of a whole new era in computing, intelligence, and human-machine interaction. This biological AI represents a paradigm leap, rather than only a little advancement. Synthetic biological intelligence has the potential to be the cornerstone of a new digital universe that thinks, adapts, and changes exactly like humans. It might revolutionize medication discovery and disease modeling, create hyper-intelligent robots, and push the boundaries of human cognition. AI's future is living and not simply made of silicon and code. The future of AI is here, and it's unlike anything we've ever seen. Is this the next step in evolution, or the start of something we can't control? Let me know your thoughts in the comments. If you found this mind-blowing, like and subscribe for more deep dives into cutting-edge tech, and turn on notifications so you don't miss what's coming next. Trust me, this is just the beginning. Let's explore the future together.